This video will be on the genus Placazoa. Will be by Nick, Zach, Lucien, and Anthony. The specific Placazoa species that we will be focusing on is the Trichoplax adherens. So the beginning characteristics of the Placazoa. It's the smallest amount of DNA that's ever been measured for any type of animal. Their bodies are comprised of 4 types of cells, and they only have a few thousand cells in total. The 4 types of cells are cylindrical cells, gland cells, cover cells, and fiber cells. For movement, Placazoa either glide using their cilia or change their shape into an amoeba-like substance in order to move. The drawing of the Trichoplax adherens, you can see that it looks like a mossy or a hairy plate substance, and it also has darker spots in certain areas. The 3 different types or main types of the Placazoa include the Trichoplax adherens, the Hoylungia Hong Kong genus, and the Polyplactoma mediterranea. For the characteristics of the Trichoplax, it's up to 2 millimeters in size. It has an irregular hairy plate shape, and it's also changing its shape constantly. It doesn't have any specific organ or muscle cells, and it doesn't have any symmetry, which can be seen in the previous picture. Characteristics of the Hoylungia: the organism is very similar to the Trichoplax. However, there's a genetic difference in the mitochondria, which is one of the main differences. And also, its habitat is the mangrove swamps of Hong Kong. For the Polyplactoma mediterranea, one of the biggest differences is that this Placazoa can grow up to 10 millimeters. And then another big difference is the expression of the mitochondrial genome, which it has some of the unique gene expressions which are for Placazoas are not expressed in the Polyplactoma mediterranea.
Trichoplax are found in the tidal zones of tropical seas and oceans, crawling on their substrates. They are discovered accidentally in an aquarium feeding on algae. Their diet consists primarily of green algae, cyanobacteria, and waste from other organisms. Propelled by their cilia, Trichoplax scour its habitat for food. When found, the Trichoplax will enclose its food, on which it will secrete digestive enzymes from gland cells. Once broken down, silated cells consume the broken down nutrients. The digestive process can only be extracellular but also phagocytic, with nutrients being consumed by phagocytosis through intercellular gaps on the surface of the Trichoplax. Because Trichoplax have no stomach nor mouth nor anus, they're neither protostomes nor deuterostomes. Because they lack veins, blood, muscle, and so many other regular animal components, they are the simplest known animal. While Trichoplax have been observed to reproduce asexually, they are believed to also reproduce sexually. Scientists have examined Trichoplax with genes inherited from 2 parents, but they've never been caught in the act. Trichoplax also contain many genes for such a simple animal with only 6 different cell types, but it's been theorized that the many unknown genes code a larval life stage. Trichoplax begin life as a larvae, then once they encounter a rock base, they develop into the adult we recognize as a Trichoplax. Although simplistic, Trichoplax contain a deadly venom that they use to paralyze potential predators, such as snails. Some Trichoplax genes are relatively similar to the venom genes of common venomous snakes, such as American copperheads and West African carpet vipers. So while Trichoplax are small, their poisons are a lethal defense against similar-sized incoming predators, such as snails.
Here's a phylogenetic graph based on IRM presynaptic calcium channel protein homologs for early metazoans. However, our taxa is one of the 9 phylum without fossil record, so I couldn't find geologically how old Placazoa are. We made our own data matrix for our organism, so we recorded different characteristics, resulting in a different phylogenetic tree. The data matrix that we made was based off of visual observations, such as cephalization, digestion, and body cavities, which are all not based in genetics, like this phylogenetic tree here, so it results in differences.
Now we'll be looking at the two phylogenetic trees made on Mesquite on the phylums that we've studied, as well as phylum Placazoa, our phylum. The two trees are similar in the way that phylum Porifera is the outgroup, but they differ in one being a phylogenetic tree made from the data matrix that we got info for, and this data is based on characteristics, while the 16S tree, phylogenetic tree, is based off of 16S rRNA, which makes it technically more accurate, while the data matrix in theory should be correct, but is not. And we can see, going into closer detail, that there are some differences. In the 16S tree, phylum Placazoa is most closely related to phylum Nadaria and phylum Porifera, while in the data matrix tree, phylum Placazoa is most closely related to phylum Annelida. And you can see some differences in the data matrix phylogenetic tree are echinodermata could be interrupting this and making the skew in the phylogenetic tree, as well as phylum Chordata and mollusca are also very different in this data matrix tree compared to the 16S tree, and you can see them circled in blue, the differences. So that was the end of our phylogenetic trees, and here's just a little photo of a Placazoa, and that is the end of our video. So just thank you for watching. Thank you to the LMU biology department, and thank you to Dr. Biddick and our TA Siraj for all the help.